Good evening. Welcome back to my daily lectures on biology and chemistry for those aspirants aspiring to appear for the NEET biology, NEET exams, and the All India Institute of Medical Sciences, the CSIR Life Sciences, and the National Eligibility Test. Right, continuing with the portions that we finished yesterday. There's an end portion which I'd like to finish right now and then continue to the next topic. So welcome back all of you. Listen carefully, please take screenshots and print out as you might wish to and make additional notes. And also make notes of the Q&A that follows every month and weekends if possible, right? So towards the end of the portions, I was talking about the lysosomes and Gaucher's disease and various aspects like that. So to the end portion of it, we have a topic on endocytosis and lysosomes formation and phagocytosis and autophagy. Once that is over, we go on to the next topic, right? So endocytosis and lysosome formation. One of the major functions of lysosomes is the digestion of material taken up from outside the cell by endocytosis. However, the role of lysosomes in the digestion of material taken up by endocytosis relates not only to the function of lysosomes, but also to their formation. In particular, lysosomes are formed by the fusion of transport vesicles budded from the trans-Golgi network and endosomes, which contain molecules taken up by endocytosis at the plasma membrane. The formation of lysosomes thus represents an intersection between the secretory pathway through which lysosomal proteins are processed and the endocytic pathway through which extracellular molecules are taken up at the cell surface. Material from outside the cell is taken up in clathrin-coated endocytic vesicles, which bud from the plasma membrane and then fuse with early endosomes. Membrane components are then recycled to the plasma membrane, and the early endosomes gradually mature into late endosomes, which are the precursors to lysosomes. One of the important changes during endosome maturation is the lowering of internal pH to about 5.5, which plays a key role in the delivery of lysosomal acid hydrolases from the trans-Golgi network. As discussed earlier, acid hydrolases are targeted in lysosomes by mannose-6-phosphate residues, which are recognized by mannose-6-phosphate receptors in the trans-Golgi network and packaged into clathrin-coated vesicles. Following removal of the clathrin coat, these transport vesicles fuse with the late endosomes, and the acidic internal pH causes the hydrolases to dissociate from the mannose-6 receptor. The hydrolases are thus released into the lumen of the endosome, while the receptors remain in the membrane and are eventually recycled to the Golgi. Late endosomes then mature into lysosomes as they acquire a full complement of acid hydrolases, which digest the molecules originally taken up by endocytosis. Phagocytosis and autophagy. In addition to degrading molecules taken up by endocytosis, lysosomes digest material derived from two other routes, phagocytosis and autophagy. In phagocytosis, specialized cells such as macrophages take up and degrade large particles including bacteria, cell debris, and aged cells that need to be eliminated from the body. Such large particles are taken up in phagocytic vacuoles, phagosomes, and which then fuse with lysosomes, resulting in digestion of their contents. The lysosomes formed in this way, phagolysosomes, can be quite large and heterogeneous, since the size and shape is determined by the content of material that is being digested. Lysosomes are also responsible for autophagy, the gradual turnover of the cell's own components. The first step of autophagy appears to be the enclosure of an organelle, example, a mitochondrion, in membrane-driven from the ER. The resulting vesicle is an autophagosome, then fuses with the lysosome, and its contents are digested. So that's it about phagocytosis and autophagy. We're going on to the next topic. It's called peroxisomes as topic B number B5. Peroxisomes are ubiquitous organelles and eukaryotes that participate in the metabolism of fatty acids and other metabolites. Peroxisomes have enzymes that rid the cell of toxic peroxides. They have a single lipid bilayer member that separates their contents from their cytosol, the internal fluid of the cell, and contain membrane proteins critical for various functions, such as importing proteins into the organelles and aiding in proliferation. Like lysosomes, peroxisomes are part of the secretory pathway of a cell, but they are much more dynamic and can replicate by enlarging them and then dividing. Peroxisomes were identified as cellular organelles by the Belgian cytologist Christian de Duve in 1967 after they had been first described in a Swedish PhD thesis a decade later. Occurrence and evolution. Peroxisomes are found in virtually all eukaryotic cells. Peroxisomes contain enzymes for certain oxidative reactions, like the beta oxidation of very long-chain fatty acids. Prokaryotes, like peroxisomes, the enzymatic content of peroxisomes varies across species, but the presence of certain proteins common to many species has been used to suggest an endosymbiotic origin, that is, peroxisomes evolved from bacteria that invaded larger cells as parasites, and very gradually evolved a symbiotic relationship. However, this view has been challenged by recent discoveries. For example, peroxisome-less mutants can restore peroxisomes upon introduction of the wild-type gene, and peroxisomes have been observed to be formed from the endoplasmic reticulum. An evolutionary analysis of the peroxisomal proteome found homologies between the peroxisomal import machinery and the AIIMS pathway in the endoplasmic reticulum, along with a number of metabolic enzymes that were likely recruited from the mitochondria. These results indicate that the peroxisome does not have an endosymbiotic origin. Instead, it likely originates from the ER. And its proteins were recruited from pools existing within the primitive eukaryote, as quoted in the science textbook of Biozone. That's a reference material, actually. Function. Peroxisomes contain oxidative enzymes such as catalase, D-amino acid oxidase, and uric acid oxidase. Certain enzymes within the peroxisome, by using molecular oxygen, remove hydrogen atoms from the specific organic substrates, labeled as R, in an oxidative reaction, producing hydrogen peroxide, H2O2 itself, toxic. RH2 plus O2 gives R plus H2O2. Catalase, another enzyme of the peroxisome, in turn uses this hydrogen peroxide, or H2O2, to oxidize the other substrates, including phenols, formic acid, formaldehyde, and alcohol, by means of the peroxidation reaction. H2O2 plus RH2 gives R plus 2H2O, thus eliminating the poisonous hydrogen peroxide in the process. This reaction is important in liver and kidney cells, where the peroxisomes detoxify various toxic substances that enter the blood. About 25% of the ethanol we drink is oxidized to acetaldehyde in this way. In addition, when excess H2O2, let me just read that again. This reaction is important in liver and kidney cells, where the peroxisomes detoxify various toxic substances that enter the blood. About 25% of the ethanol we drink is oxidized to acetaldehyde in this way. In addition, when excess H2O2 accumulates in the cells, catalase converts it to H2O through this reaction, so 2 hydrogen peroxide gives 2H2O plus O2. A major function of the peroxisome is the breakdown of fatty acid molecules in a process called beta oxidation. In this process, the fatty acids are broken down to 2 carbons at a time, converted to acyl-CoA, which is then transported back to the cytosol for further use. In animal cells, beta oxidation can occur in the mitochondria. In yeast and plant cells, the process is exclusive for the peroxisome. The first reaction in the formation of plasmalogen in animal cells also occurs in peroxisomes. Plasmalogen is the most abundant phospholipid in myelin. Deficiency of Plasmalogens causes profound abnormalities in the amyelination of nerve cells, which is one of the reasons that many peroxisomal disorders lead to neurological disease. Peroxisomes also play a role in the production of bile acids and proteins. In higher plants, peroxisomes contain also a complex battery of antioxidative enzymes such as superoxide dismutase, the components of the ascorbic glutathione cycle, and the NADP dehydrogenases of the pentose phosphate pathway. It has been demonstrated the generation of superoxide O2 negative and nitric oxide NO radicals, they are also involved in photorespiration. In plants, special type of peroxisomes termed as glyoxysome carry out glyoxylate cycle, in which lipids stored in seed are converted to carbohydrates and thus provides source of nutrition for developing embryos. Peroxisomes also play a role in the production of bile acids and proteins. In higher plants, peroxisomes contain also a complex battery of antioxidative enzymes such as superoxide dismutase, the components of the ascorbic glutathione cycle, and the NADP dehydrogenases of the pentose phosphate pathway. It has been demonstrated that the generation of superoxide O2 negative and nitric oxide NO radicals, they are also involved in photorespiration. In plant special type of peroxisomes termed as glyoxysome carry out the glyoxylate cycle in which lipids are stored seed and are converted to carbohydrates and thus provide source of nutrition for developing embryo. Protein import. Proteins are selectively imported into peroxisomes since the organelles contain no DNA or ribosomes and thus have no means of producing proteins. All of the proteins must be imposed across the membrane. It is believed that necessary proteins enter through the endoplasmic reticulum during biogenesis as well as through member proteins. A specific protein signal, PTS, or peroxisome targeting signal, of 3 amino acids at the C terminals of many peroxisomal proteins, signals the membrane of the peroxisome to import them into organelles. Other peroxisomal proteins contain a signal at the N terminus. There are at least 32 known peroxisomal proteins called peroxins, which participate in the process of importing proteins by means of ATP hydrolysis. Proteins do not have to unfold to be imported into the peroxisome. The protein receptors, the peroxins PEX5 and PEX7, accompany their cargoes containing a PTS1 or PTS2, respectively, all the way into the peroxisome, where they release the cargo and then return to the cytosol, a step named recycling. Overall, the import cycle is referred to as the extended shuttle mechanism. Evidence now indicates that ATP hydrolysis is required for the recycling of receptors to the cytosol. Also, ubiquitination appears to be crucial for the export of PEX5 from the peroxisome to the cytosol. Little is known about the import of PEX7, although it has helper proteins that have been shown to be ubiquitinated. Deficiencies. Peroxisomal disorders are a class of conditions that lead to disorders of lipid metabolism. One well-known example is Zellweger syndrome. Peroxisomes matrix proteins are synthesized on free ribosomes in the cytosol, and that these proteins are imported post-translationally in pre-existing peroxisomes. The next portion would be B6 vacuoles, right? Vacuoles or vacuoles, as you might call it. Vacuoles are found in the cytoplasm of most plant cells and subanimal cells. Vacuoles are membrane-bound compartments with some eukaryotic cells that can serve as a variety of secretory, excretory, and storage functions. Vacuoles and their contents are considered to be distinct from the cytoplasm, and are classified as agastic. According to some authors, vacuoles are especially conspicuous in most plants. Functions. In general, vacuole functions include also removing unwanted structural debris, isolating materials that might be harmful or a threat to the cell, containing waste products, maintaining internal hydrostatic pressure or turgor pressure within the cell, maintaining an acidic internal pH, containing small molecules, exporting unwanted substances from the cell. Vacuoles also play a major role in autophagy, maintaining a balance between biogenesis, production, and degradation of turnover of many substances and cell structure. Vacuoles store food and other materials needed by a cell. They also aid in destruction of invading bacteria or of misfolded proteins that have begun to build up within the cell. The vacuole is a major part of the plant and animal cell. Protists. Some protists and macrophages use food vacuoles as a stage in phagocytosis. The intake of large molecules, particles, or even other cells by the cell for digestion, they are also called storage sacs. A contractile vacuole is used to pump excess water out of the cell to reduce osmotic pressure and keeps the cell from bursting, which is referred to as cytolysis or osmotic lysis. Budding yeast. In budding yeast cells, vacuoles act as storage compartments of amino acids and detoxification compartments. Under conditions of starvation, proteins are degraded in vacuoles. This is called autophagy. First, cytoplasm, mitochondria, and small organelles are covered with multiplex plasma membranes called autophagosomes. Next, the autophagosomes fuse the vacuoles. Finally, the cytoplasms and the organelles are degraded. In a vacuole lining, vacuole of budding yeast, black particles sometimes appear, called a dancing body. The dancing body moves actively in the vacuole and appears and disappears within 10 minutes to several hours. In previous research, it was suggested but not proven that the main component of the dancing body is polyphosphate acid, but the main component has been determined to be crystallized sodium polyphosphate, and its function has been studied. It is thought that its function to supply and store phosphates in budding yeast cells. Plants. Most mature plant cells have one or several vacuoles that typically occur more than 30% of the cell's volume. That can occupy as much as 90% of the volume for certain cell types and conditions. A vacuole is surrounded by a membrane called the tonoplast. This vacuole houses large amounts of a liquid called cell sap, composed of water, enzymes, inorganic ions like K plus and Cl, that's chlorine, salts such as calcium, and other substances, including toxic byproducts removed from the cytosol to avoid interference with metabolism. Toxics present in the vacuole may also lead to protect some plant from predators. Transport of protons from cytosol to vacuole aids in keeping cytoplasmic pH stable or making the vacuolar interior more acidic, allowing degradative enzymes to act. Although having a large central vacuole is the most common case, the size and number of vacuoles may vary in different tissues and the stages of development. Cells of the vascular cambium, for example, have many small vacuoles in winter and one large in summer. Aside from storage, the main role of the central vacuole is to maintain turgor pressure against the cell wall. Proteins found in the tonoplast control the flow of water into and out of the vacuole through active transport, pumping potassium K plus ions from into and out of the vacuole interior. Due to osmosis, water would diffuse into a vacuole, placing pressure on the cell wall. If the water loss leads to a significant decline in turgor pressure, the cell wall will plasmolyze. Turgor pressure exerted by vacuoles is also helpful for cellular elongation. As the cell wall is partially degraded by the action of oxygens, the less rigid wall is expanded by the pressure coming from within the vacuoles. Vacuole can help some plant cells to reach considerable size. Another function of a cell vacuole is that it pushes all contents of the vac cells of cytoplasm against the cellular membrane and thus keeps the chloroplasts closer to light. The vacuole also stores the pigments in flowers and fruits. Animals is the next section. Vacuoles in animals are a part of the process of exocytosis and endocytosis. Exocytosis is the extrusion process of proteins from the Golgi apparatus, initially either intersecretory granules, where processing of prohormones to the mature hormones occurs before exocytosis, and also allows the animal's cell to rid waste products. Endocytosis is the reverse of exocytosis. There are various types. Phagocytosis, cell eating, is the process by which bacteria, dead tissue, or other bits of material visible under the microscope are engulfed by cells. The material makes contact with the cell membrane, which then invaginates. The invagination is pinched off, leaving the engulfed material in the membrane enclosed vacuole and the cell membrane intact. Pinocytosis, cell drinking, is essentially the same process, the difference being that the substance ingested are in solution not visible under the microscope. Hydropic vacuolar changes are of importance of identifying various pathologies such as the reversible cell swelling in renal tubules caused by hyperperfusion of the kidneys during the open heart surgery. Plastids. Plastids are major organelles found in plants and algae. Plastids often contain pigments used in photosynthesis, and the types of pigments present can change or determine the cell's color. Plastids are also the size of manufacture at the site of manufacture and storage of important chemical compounds used by the cell. Plastids in plants. Plastids are responsible for photosynthesis, storage of products like starch, and for the synthesis of many classes of molecules such as fatty acids and terpenes, which are needed as cellular binding blocks and or for the function of the plant. Depending on their morphology and function, plastids have the ability to differentiate or to redifferentiate between these and other forms. All plastids are derived from proplastids, formerly eoplastids, or eo, and dawn early, that's the meaning of eo, which are present in the meristematic regions of the plant. Proplastids and young chloroplasts are commonly divided, but more mature chloroplasts also have this capacity. In plants, plastids may differentiate into several forms depending upon their function they need to play in the cell. Undifferentiated plastids proplastids may develop into any of the following plastids: chloroplast for photosynthesis, see also eschioplast and the predecessors of chloroplasts, chromoplast for pigment synthesis and storage, leucoplast for monoterpene synthesis, leucoplast sometimes differentiated into more specialized plastids, amyloplast for starch storage, statoliths for detecting gravity, eleoplast for storing fat, proteinoplasts for storing and modifying protein, chloroplasts for photosynthesis, see also eschioplast, the predecessors of chloroplasts, chromoplast for pigment synthesis and storage, leucoplast for monoterpene synthesis, leucoplast, sometimes differentiated into more specialized plastids. Amyloplast for starch storage, statoliths for detecting gravity, eleoplast for storing fats, proteinoplast for storing and modifying protein. Each plastid creates multiple copies of the circular 75-20-250 kilobases plastid genome. The number of genome copies per plastid is flexible, ranging from more than 1,000 and rapidly dividing cells, which generally contain 2 plastids, 200 or fewer in mature cells where plastid divisions has given rise to a large number of plastids. The plastid genome contains about 100 genes encoding ribosomal and transfer ribonucleic acids, RNAs, rRNAs, and tRNAs, as well as proteins involved in photosynthesis and plastid gene transcription and translation. However, these proteins only represent a small fraction of the total protein setup necessary to build and maintain the structure and function of a particular type of plastid. Nuclear genes encode the vast majority of plastid proteins, and the expression of plastid genes and nuclear genes is tightly co-regulated to allow proper development of plastids in relation to cell differentiation. Plastid DNA exists as large protein DNA complexes associated with the inner envelope membrane and called plastid nucleoids. Each nucleoid particle may contain more than 10 copies of the plastid DNA. The proplastid contains a single nucleoid located in the center of the plastid. The developing plastid has many nucleoids localized at the periphery of the plastid bound to the inner envelope membrane. During the development of proplastids, chloroplasts, and when plastids convert from one type to another, nucleoids change in morphology, size, and location within the organelle. The remodeling of nucleoids is believed to occur by modifications to the composition and abundance of nucleoid proteins. Many plastids, particularly those responsible for photosynthesis, possess numerous internal membrane layers. In plant cells, long thin protuberances called stromules sometimes form and extend from the main plastid body into the cytosol and interconnect several plastids. Proteins and presumably smaller molecules can move within stromules. Most cultured cells that are relatively large compared to other plant cells have very long and abundant stromules that extend to the cell periphery. Plastids in algae. In algae, the term leucoplast is used for all unpigmented plastids. The function differ from the leucoplast in plants. Eschioplast, myeloplast, and chromoplast are plant-specific and do not occur in algae. Algal plastids may also differ from the plant plastids in that they contain pyronoids. Inheritance of plastids. Most plants inherit the plastids from only one parent. Angiosperms generally inherit plastids from the mother, while many gymnosperms inherit plastids from the father. Algae also inherit plastids from only one parent. The parent plastid DNA of the other parent is thus completely host. Is completely lost. In normal intraspecific crossings resulting in normal hybrids of one species, the inheritance of plastid DNA appears to be quite strictly 100% uniparental. In interspecific hybridizations, however, the inheritance of plastids appears to be erratic. Although plastids inherit mainly maternally in interspecific hybridizations, there are many reports of hybrids of flowering plants that contain plastids of the other. Origin of plastids. Plastids are thought to have originated from entosymbiotic cyanobacteria. They developed around 1500 MYA and allowed eukaryotes to carry out oxygenic photosynthesis. Due to these split up into three evolutionary lineages, the plastids are named differently: chloroplasts in the green algae and red plants, rhodoplasts in red algae, and cyanols in the glaucophytes. The plastids differ by their pigmentation and also in ultrastructure. The chloroplasts, example, have lost all phycobilisomes, the light harvesting complexes found in cyanobacteria, red algae, and glaucophytes, but only in plants and in closely relative green algae contain stroma and grana thylakoids. The Glucosylcephine plastid, in contrast to chloroplast and the rhodoplast, is still surrounded by means of cyanobacterial cell wall. All these primary plastids are surrounded by two membranes. Complex plastids start by secondary endosymbiosis when a eukaryote engulfs a red or green algae and retains the algal eukaryote algal plastid. I repeat, when a eukaryote engulfs a red or green algae and retains the algal plastid, which is typically surrounded by more than two membranes. In some cases, the plastids may be reduced in their metabolic and/or phagosynthetic capacity. Algae with complex plastids derived from secondary endosymbiosis of a red algae include the heteroconchs, haptophytes, cryptomonads, and most dinoflagellates, for example, rhodoplasts. Those that endosymbiotic green alga, including the euglenids and chlorarachnophytes, equal to chloroplasts. The Epicomplexer, a phylum of obligate parasitic protozoa, including the causatives, agents of malaria, Plasmodium spp, Toxoplasmosis, Toxoplasma gondii. Many of the human and animal diseases also have a complex plastid, although this organelle has been lost in some epicomplexans, such as Cytosporidium parvum, which causes cryptosporidiosis. The apicoplast is no longer capable of synthesis, but is an essential organelle and a promising target for intraparasitic drug development. Some dinoflagellates take up algae as food and keep the plastid of the digested algae to profit from the photosynthesis. After a while, the plastids are also digested. These captured plastids are known as kleptoplastids.